Hello, Dr. Cisse covering anatomy and physiology. Today we'll talk about chapter 4, genetics and cell functions. Now we'll talk about chromosome, DNA, and how DNA is coiled to be preserved and conserved in the nucleus of the cell. So let's talk about DNA and RNA in general. In this class, we talk about 4 macromolecules, right? Organic molecules: proteins, lipids, carbohydrates, and nucleic acids. So today we'll talk about nucleic acids, which are what? DNA and RNA. We'll talk about the DNA structure, RNA structure, the function of mRNA, why we need mRNA, what is mRNA, why we need it, and then the tRNA, what is tRNA, where is it located, what does it look like, and then how do we make stuff in our body, how do we make proteins in our body. So the question you're going to be able to answer in this video, you need to be able to tell me 10 differences between DNA and RNA. What are the differences, right? The structure, the function, how do they do things? And then we'll talk about transcription and translation. You have to be able to tell me 5 or even 10 differences between transcription and translation. So what does DNA stand for? Deoxyribonucleic acid, DNA. It is the long thread-like molecule with uniform diameter, but a varied length, right? So DNA can be different length. So in human, we have 46 DNA molecules that we call chromosomes, and they're located in the nucleus of most human cells. So the average length is 2 inches. So we say that DNA and other nucleic acid, which are for other RNA, are a polymer of nucleotide. It means it's a bunch of things together, bunch of nucleotides following each other. It's called polymer. They have a sugar, phosphate group, and nitrogenous base. So the sugar and phosphate group and base, the 3 together represent the molecule of DNA or the nucleic acid. So we have 1 sugar for DNA. For example, the sugar in DNA is called deoxyribose. And we have 1 phosphate group and 1 nitrogenous base. And that base could be a purine, which is adenine or guanine, or pyrimidine, which is cytosine, thymine. In RNA, you're going to have uracil, right? So in DNA base, we have A, T, C, G. What is the nitrogenous base, right? It's a base that 2 bases that are united by hydrogen bond. Remember when we talk about hydrogen bond in water molecule? The bond between 2 different water molecules. Here, the 2 strands of DNA are attracted. They're not even linked. They're just attracted by hydrogen bond. So every time you have an A, you have a T on the other side. Every time you have a C, you have a G, right? So we have a law called the law of complementarity base pairing. It means anytime you have an A, it's connected to a T. Anytime you have a C, it's connected to a G. So this is what the nucleotide look like, right? Adenine, deoxyribose, which is a sugar, and the phosphate group, PO4. And we said that DNA is the double helix. It means it's like a stair. It has 2 sides, and the 2 sides are connected. You can see the H bond here between the A and the T and the C and G. You see it in detail here. So it looks like it's kind of coiled, right? Coiled double-strand DNA, complementary to each other. Now let's talk about RNA. What is RNA? RNA stands for ribonucleic acid, RNA. So DNA and RNA are both nucleic acid. It is smaller molecule that resemble DNA. So one difference between DNA and RNA, RNAs are smaller. They can have a less than 100 or just over 10,000 base pair molecules. And DNA can have millions. So we have 3 important RNA for protein synthesis: mRNA, rRNA, and tRNA, right? So they are not double helix. That's another difference, right? And then the ribose replaces the deoxyribose as sugar. So they have 2 different kind of sugar also. And uracil replaces thymine. So instead of having T in RNA, we have U. That's another difference. And the RNA function in the cytoplasm, not in the nucleus. So that's another difference, right? So let's talk about the gene in general. What is the gene versus the genome? A gene is a segment of a DNA, right? The reason why we talk about gene in the DNA, because we understand what is the DNA, but a little part of the DNA is called a gene. Why? Because that little part will be coding for the synthesis of a specific protein, right? If you take all the genes together, it's called the genome. Inside the cell, in the middle, in the nucleus of the cell, we have all the DNA molecules are mixed up, and this is called chromatin. When the cell is not dividing, we can see it. It's kind of just a mixture, just like a spaghetti with some kind of milk or something, right? In a non-dividing cell. But when the cell started to divide, the DNA will be dividing. They're going to be like forming structure that you can identify. Those structures are called chromosomes. So chromosomes are just the same as DNA, just with a very specific shape. For instance, it's a chromosome. Why this is important? When the cell is dividing, remember, the DNA has to be replicated. It has to be double up. I mean, in order for you to be very organized, you want to have a specific shape so that when you divide, each part of the chromosome can be divided between the two daughter cells. So what is the genetic code? It's a system that enables these 4 nucleotides, remember, CGAT, to code for amino acid sequence of all proteins. It's just like a code in your body that can determine which amino acid, which protein is being made. And it comes in a triplet. It means that every mRNA will have 3 pieces of those nucleotide that will be making a specific amino acid. And many amino acids will be the protein. So basically the way your body makes stuff, makes protein, depends on which code is being used. And the combination of the code determine which amino acid is being made, and the combination of amino acid determine which protein is being made. So what is transcription then? When you take the DNA in the nucleus, it has the code, right? And there has to have a machine that can read that code to transcribe, to write in another language. So transcription is just copying a genetic instruction from the DNA to mRNA. Now, RNA polymerase is an enzyme that has to bind to the DNA to be able to make the mRNA. One example, let's say, for example, your grandmother has a recipe to make pizza. You're going to be RNA polymerase. Your grandmother is the DNA. So you go to your grandma and say, Grandma, I need the recipe to make pizza. I mean, your grandmother has that information in her brain. All you do is write it down on a piece of paper. So she might say, for example, you've taken dough, you mix it with milk, and then you mix it with the salt and egg. So everything she tells you, you write it down. In biology, your grandmother is the DNA. You are RNA polymerase. And the recipe you write on the piece of paper is the mRNA. Okay? Let me go back again, right? So your grandmother has the information to make pizza. So she's the DNA in the nucleus. You are the RNA polymerase. Why? Because you want to go to her to get a recipe. And then you have to write that recipe on the piece of paper. So the piece of paper and the recipe will be the mRNA. It's just a message that you're taking to go make the pizza. We're not at the pizza making yet, but at this point you have a message, right? So it means that certain DNA-based sequences signals start. It means the beginning of the recipe. Let's say the beginning will be, for example, making pizza. So that is ta-da-da, for example. So if you say making pizza, we know you're making pizza. Because you could be making bread also. You could be making something else. So RNA polymerase, you opens up the DNA helix, meaning you open your grandmother's brain and read the stuff, right? You read it and take a note. The note you're taking is called mRNA. So what is transcription then? Transcription is the making mRNA from DNA. It's making a recipe from your grandmother's brain, for example, right? So you're copying genetic instruction from DNA to mRNA. You copy the pizza making recipe from your grandmother's brain to your piece of paper. So you are the RNA polymerase. You're an enzyme. Remember, enzymes are proteins, right? you bind to DNA. So you actually jump on your grandmother's shoulder and take the information from your brain and write that on the piece of paper that will be the mRNA, right? Certain DNA base sequences signal start. For example, Tadada, we talk about this. RNA polymerase open up DNA helix. What do? You open your grandmother's brain to take the information, right? So every time you see a C on the DNA, for example, your grandmother, every time she say the C, you write a G on that piece of paper. Anytime she say G, you write a C on the piece of paper. Anytime she say T, you write A. Anytime she say A, you write O, U, right? So basically what I'm trying to say is you copying something, but you don't copy specifically. It's just a code, right? So polymerase, RNA polymerase, does the same thing. It just takes the information from the grandmother's brain, which is the DNA, to make recipe, which is the mRNA, by using specific code. But it does not write specific what the grandmother has, but you know every time you see a C, it has to be a G on your piece of paper. So when somebody reads a G, they can predict what's in your grandmother's brain because your G will be her C, basically, right? All right. So, but what is alternative splicing of mRNA? After you make all the recipe, you're gonna have some stuff you wrote that don't make any sense. You're gonna be like, she said bread, you know. So when you actually making the bread, you don't use all the words in the recipe, right? Some of them you have to cut them out because they're useless. You just wrote them so you can be like a little point, maybe exclamation point, or maybe a little. So you don't use those because those are not going to be used for the recipe. You only use for the bread, the dough, the egg, the milk, stuff like that. So at the end, at the bottom here, protein, you're making your pizza, right? The beginning is the DNA here on top. This is like your grandmother's brain. You take polymerase, you are the polymerase, to make the mRNA, which is the recipe. Then you have to cut it in pieces. Why? Because some part of the recipe do not make any sense. Then each piece, you kind of make a bread. This one is you make a pizza. Maybe another one you kind of making bread. The other one you're making cake. So the bread and cake and pizza are the protein that you're making. The making of the protein from mRNA is called translation. Translation. Okay? So translation is the making of the protein from the mRNA, making bread out of the recipe. So the process that convert the language of nucleotide, basically what you write on the recipe, you kind of go to your kitchen and actually making the bread with pieces of maybe the recipe. For example, you have the dough, you have the milk, you have the egg. Those are amino acids so that you can build your pizza, you can make your protein. So the three main participants in the translation are the mRNA. You have to have the recipe that carries the code that has the information from the nucleus, which is the grandmother's brain, to the cytoplasm into the kitchen. The kitchen will be the cytoplasm in this case. So the mRNA has protein cap that is a recognized site for ribosome. Now, what is the ribosome? The ribosome will be like your kitchen oven, right? So you're gonna go to the oven and make your bread now. So inside the oven, you're gonna have to have pieces of building blocks of your pizza. Who will give that to you? It's going to be your transfer RNA. What is transfer RNA? Transfer RNA are just nucleic acid, because they're RNA, right? That will take amino acid, the building block of your pizza, to give it to you, right? Inside your ribosome, which will be your kitchen, your oven, right? So it will use those code. Anytime you have the recipe, let's say for example, you have three pieces. You have, for example, 3 nucleotide, 3 pieces on the recipe. Your tRNA will know that it has to be this amino acid. It has to be this particular part of the bread to give it to you so you can build up your pizza. What is the ribosome? Organelles that read the message. It means it is the oven that will tell you how to make the pizza based on the information you got from your grandmother that you wrote on the piece of paper called the mRNA. The messenger RNA is just a message to make the protein. Where do you find the ribosomes? Where do you find your oven? In your kitchen. The kitchen will be the cytosol. Remember the difference between cytoplasm? Cytoplasm will be all the organelles in the cell with the liquid, the gel type of liquid called the cytosol. So the cytosol is the liquid part of it. It's going to be on the rough ER. Why is the rough ER called rough ER? Because it has a ribosome. Why do you need the ribosome? Because the ribosomes are the machinery that make protein in your body. It's just like your oven in your kitchen that will bake your bread in your house. So it has a large subunit and small subunit, where each subunit is made of several enzyme and ribosomal RNA molecule. So inside your oven, which is your ribosome, you have ribosomal RNA. You have pieces of RNA there too. You have a lot of enzyme. It's like a machine, it's like a kitchen. You have different things to make, basically bake your bread. In this case, to make your protein. How does it happen? The mRNA goes through the ribosome, just like you put your recipe into your oven to get pieces of building block of your pizza. In this case, the building block will be the amino acid to give you protein, which will be the end product, the pizza. We have steps in translation. Translation means what? Taking the mRNA to make the protein. Take the recipe to make the pizza. You have initiation, the beginning, elongation when you're adding, and termination to end the process. What is the initiation? It means that the leader sequence in mRNA binds to small ribosomal subunit. So initiator tRNA bearing methionine pairs with the start codon. I'm going to explain this. Larger ribosomal subunit join the complex, and the now fully formed ribosome begin raiding the base. It means that you're actually building your oven to make your pizza. The oven is the ribosome. It's like the door of the oven and the oven itself are coming together to hold the recipe and to bring in all the building block, all the pieces that you need to make the pizza with. For example, in the recipe, if you say dough, you bring the dough. If it's say egg, you bring the egg. Who brings the egg? The tRNA, right? The tRNA bring the pieces of the building block. How? When it's adding up, it's called elongation. Every time you say dough, the tRNA brings the dough. Every time you say egg, the tRNA brings the egg. Every time you say so, they bring it. The process of bringing and adding is called the elongation. And at the end, when you finish at the end of recipe and the mRNA, it says termination, stops making the pizza. So what is the tRNA? It is the small RNA molecule, right? Coiled on itself to form an angular L shape. It's different than DNA. You can tell differences between DNA and RNA here. So what does the tRNA do? It picks up free amino acid in cytosol. It picks up free building blocks of your pizza in your kitchen to bring them to the oven to build the pizza. And it takes energy. Of course, you have to plug your oven to make it happen, or you have to use gas. You have to use energy to bake your pizza. So it has to use ATP in the cell. So this is what the tRNA looks like. Just imagine, it has, for example, this one, it has CCA. It has 3 nucleotides, and each 3 nucleotide is corresponding to 1 amino acid. Every time your tRNA sees on your recipe CCA, it knows it has to bring something. For example, it has to bring the egg or the dough to build the pizza. Basically, the tRNA is like somebody working in your kitchen, helping you to bring pieces of your building block of your pizza in your oven, which is the ribosome, to make your pizza, which is the protein, the end product you're trying to make. So what are the process of translation? So translation, you see here, this is the ribosome. You see the ribosomes here? This is like your oven. And then you see the tRNA bring in the different amino acid to add based on the message on the mRNA, which is your recipe that you wrote from your grandmother, which is your DNA. So translation of mRNA into free or transport vesicle. Depending on whether you're going to be using the bread in your kitchen for yourself to eat it, or you're going to ship it to somebody else in another city, you have different kind of ribosome. You have a free ribosome in the cytosol that can make a protein inside the cell, or you can have ribosome bound to rough ER that will be making the protein that will be shipped. Let's say you need to make a protein to go to another city, right? It'll be shipped there. Those ribosomes will be bound into rough ER. So to review the peptide formation, peptide just means the protein making. You have the DNA, double helix, from which you will make mRNA, right? And that mRNA will go through the ribosomes with the tRNA bringing amino acid to make different amino acid that will come together to become the protein, right? So you say DNA double helix, 7 base triples on the template strand of DNA here, the corresponding codon of mRNA, right? So here you have the grandmother's brain, you make a recipe, You go to your kitchen, somebody's helping you to bring the recipe. You build different building block of the pizza. You put it together, it becomes the pizza. Okay? So translation to transport vesicle, and those are shipped out in different vesicles. Now, you would ask yourself, you know you're making proteins, right? What about other stuff you make? For example, we can make glycogen, fat, steroid, phospholipid, pigment, and other compound, but we don't use gene for them, right? We don't go to the DNA for them. Those are things that are transformed in your body from previously made stuff. So they're kind of making things from each other. For example, if you produce testosterone, you take cholesterol and then to convert to testosterone. So that's not genetically done. It's done after something is made already. So everything is not made from your gene, from your DNA. Only proteins are made from DNA. Those proteins use other stuff in your body to make things. Now, one example we can give in how we make things. Let's say we're trying to make milk, right? Prolactin is a hormone that will bind to receptors. We talked about this already. Remember, the receptor activate, and then when it activates, it go into your gene by regulatory protein. There'll be a lot of protein inside your cell that will do some job. They go bind to your DNA. In this case, tell the DNA to make something, to make casein, for example, or to make bread, for example. It goes to your DNA, which is your grandmother's brain. You are the polymerase. You unwind the DNA and you take the notes, mRNA. That mRNA is in the nucleus, right? Will go out to the ribosome on the right ER. From the mRNA, you're making the protein that will be shipped, that will be bought off to the Golgi complex. The Golgi complex is like a UPS, right? That will ship out the casein to produce in the milk. Okay? So here you can see the hormone prolactin bind to receptor. You can read this, receptor trigger activation. All this just to show you that in order for milk to be produced, something has to be excited, it has to go inside the cell to the gene for the mRNA to make a copy and go out, right? To use the tRNA inside the ribosome to make the protein, okay? So at the end, what is the gene, right? The first definition of the gene we say is a segment of DNA that carries the code for a particular protein. But if you want to go into detail, you can say a gene is an information containing segment of DNA that code for the production of a molecule of RNA, remember, mRNA, that play in a role in synthesizing one or more proteins. So making protein from mRNA, which came from a gene. A specific type of the part of the gene is called, the DNA is called a gene, right? A specific part of the DNA. So now question, can you explain 10 differences between DNA and RNA? That's going to be our video quiz. Can you explain 5 differences between transcription and translation? Think about the location. Where are they located? DNA is in the nucleus, and RNA is basically in the nucleus going out to the cytoplasm. And the tRNA is in the cytoplasm because it is on the ribosome, and the ribosome is on what? The ref ER. Then the structure, what do they look like? Remember that DNA is a double helix, but RNA is not double helix. DNA is a bigger, a lot of thousands and millions of base nucleotide, but RNA is very short. We have different type of RNA, mRNA, tRNA, and rRNA. What do they do? See, DNA has a code. It stays in the nucleus. It doesn't come out, right? And the RNA, we have different kinds. We have mRNA that takes the message, tRNA that transfer, or ribosomal RNA, rRNA that's inside the ribosome. And then how do they do what they do? DNA has a code. It kind of unwind for the polymerase U to read your grandmother's mind, to write it down as an mRNA to go out to the ribosome in order for you to make what? The protein, right? So if you look at here, the DNA here is in the nucleus. You make the template of the DNA. You copy that, right, to make the mRNA. You use the tRNA to bring amino acid to build basically your protein, right? So I hope this is going to be very helpful to you so that we can understand more. So now you know your grandmother, you know U polymerase, you know the mRNA, the recipe, you know the building block amino acid to build protein. All right, thank you very much, and I'll see you in class.